All right. Good morning, everyone here. Good evening, Randy. So hello, everyone. My name is Li Wenjiang from the Faculty of Science. So on behalf of the faculty, I would like to welcome you all to the Faculty Research Day 2021. So may I first invite Professor Song, Dean of Faculty of Science, for delivering an open remark. Professor Song, please.
Hello, everyone. Good morning to our virtual and physical participants in Hong Kong, and to Professor Randy SCHEKMAN. Good evening. Thank you all for coming to attend this Faculty Research Day. On behalf of the Faculty of Science, I'd like to welcome all of you to the Faculty of Science 2021 Research Day. Every year, the faculty organize a research day for the researchers to get together to share ideas and experience in conducting research and advancing collaboration. Last year, shortly after I joined the CUHK as the new dean, we had the first virtual research day. The first virtual day, for me, it was a great experience because it gave me the broad exposure. Even during the pandemic, I had the first glance in the broad research activity in the Faculty of Science across all the units. This year, we're launching this premier event in both physical and virtual participation, including also the keynote speaker for our research day this year. The theme for this year is advancing science through interdisciplinary and multidisciplinary research and collaboration. Another highlight of this year's Research Day is a sharing on collaborative research fund. Through interdisciplinary and multidisciplinary collaboration research, our researchers strive to advance fundamental understanding and also address the global scientific and societal challenges. The CUHK 2021-2025 strategic plan highlight the importance of interdisciplinary and multidisciplinary research. Our faculty also provided input to the CUHK strategic plan in 7 interdisciplinary and multidisciplinary areas. Our faculty, in the spirit of promoting collaborative research, last year, we selected, and through the competitive proposal process, 5 research proposals for interdisciplinary collaboration where the investigators come from 2 or more units. And some of our researchers and investigators from this collaborative fund will also share experience today. For the Faculty Research Day this year, we are very honored and extremely pleased to have most distinguished scientists and Nobel laureate, Professor Randy SCHEKMAN from the University of California at Berkeley, as our keynote speaker. Professor Randy SCHEKMAN will share his insight on collaboration and openness in basic science and research publications. I will ask my colleague, Professor Li Wenjiang, to introduce Professor SCHEKMAN in a few moments. We are also pleased to have 3 presentations by leading researchers from different units in science, including Professor Ronald Lui in mathematics, Professor Jiang Xia in chemistry, Professor Yilin Wu in physics. They will share their interdisciplinary research along the theme of research on biological problems. Over the past 10 years, the faculty has successfully developed 24 collaborative research grant projects, the CRF project, and 3 areas of excellence, the so-called AOE project, funded by the Research Grant Council of Hong Kong. We hope that the Research Day will promote the discussion among our faculty members in initiating and promoting collaborative research. Toward this objective, we have invited 7 colleagues from the Faculty of Science, Engineering, and Medicine, including Professor Manning Chen in Earth System Science, Professor Kinfa Hou in Public Health and Primary Care, Professor Yingying Wei in Statistics, Professor Sen Yang in Physics, Professor Jeremy Hui in Life Science, Professor Kevin Yip in Computer Science Engineering, as well as Professor Liwen Jiang in Life Science, to share their experience in the CRF grant application and also how they learned from the previous submissions. We hope the Research Day will serve as a platform for researchers to exchange ideas from recent research advances and also encouraging the future collaborative research grant applications. And I hope you enjoy the Research Day this year, and I will talk to you later. Thank you.
All right, thank you, Chunshan, for the opening remarks. I think now it's time for us to present the gift to the speakers. Okay, so I think Randy, have you received our present?
Yes, I have.
All right, so you don't need to.
I have it here.
All right, thank you. All right, so
I'm wearing your tie
thank you
too. I'm
again
wearing the tie you sent.
a present. Okay, so the next one is the Ronald, right? Yeah. All right, Professor Xia Jiang, please. And Professor Wu Yiling, please. Okay, Professor He Jianhui, please. All right, because Professor Wei Yin right now, she's in Shanghai, so we'll keep the present for you, okay, for now.
Thanks a lot.
And then Professor Yang Shen, please. Oh, sorry. All right, Professor Chen Manlin, sorry. Please. And Professor Yip, because you're on Zoom, we'll keep the present for you, okay?
Thank you.
And also myself, okay? Oh, okay. All right, now we would like to take a group photo, okay, for the attendance. Okay, how should we do that? So we just come out. Okay, all right. All right, welcome back. So it's a great honor for me to introduce today's keynote speaker, Professor Randy SCHEKMAN. Okay, he's the investigator of the Howard Hughes Medical Institute and university professor from University of California at Berkeley. So I don't need much introduction about Randy because he's well known, okay? Rather, I would like to mention a few things about Randy's connection with CUHK and also Hong Kong. All right, so Randy actually was one of the first invite speaker for our first culture summer course organized in CUHK in the year 2013 in August. Later on that same year, he received the Nobel Prize, okay? And also he has been very strong supporter, also collaborator for our AOE project, organelle biogenesis and function. And also he graduated from CUHK about 5 years ago. And the same year, he came all the way from Berkeley to CUHK to sign the MOU between UC Berkeley and CUHK on the BBSA study abroad program, okay? So this shows you the first 5 students we select for sending to UC Berkeley for this program. And also, I think last time when Randy visited Hong Kong was 2 years ago, before the pandemic. So that's also for the second round of the culture summer course. He is also was one of the invited speaker. So before the summer course, we have a hiking. And also in the opening, the summer course, okay, our VC was also attending this opening. And last, I would like to mention also, just 2 days ago, I captured this screen. So Randy is the selection committee chairman for the Seoul Prize in Life Science and Medicine. So he really has a lot of connection with CUHK, also Hong Kong. Okay, so we thank you again, Randy, for accepting our invitation for delivering this keynote speech. Okay, so without further delay, please join me to welcome Professor Randy SCHEKMAN.
Thank you. Thank you very much, Li Wen and Professor Song. I appreciate the opportunity. I only wish I could be there with you so I could share in this award that you've sent me. Thank you very much. And but I am wearing the tie that you also sent. So thank you for all that you've done for me over the years, Li Wen. Today I'm going to give a broader lecture on the subject of collaboration and cooperation internationally. I'm going to touch on themes that have to do with basic aspects of cell biology and just an introduction to the field that Li Wen and I are both doing our work in. I will then talk about issues in relation to open science and open publication, the open access movement, and some aspects of challenges in scientific publication, particularly in the most selective journals. And then I'm going to turn to a subject that is of more recent involvement for me, and that is an international collaborative effort to understand the basic mechanisms of Parkinson's disease. So let me It's a rather broad theme, but I hope it ties together. So let me share my slides with you and begin. Okay, so again, the importance of collaboration and openness in basic science and in research publication. Well, I'm going to start with an experiment that we have unfortunately all been subjected to for the past year plus, and that is this little beast. It's the coronavirus, and I use this as an example of many themes that I'm going to touch on. The first, of course, is this is a worldwide problem. It obeys no borders, and it requires an international effort. There have been some considerable international aspects to attempts to control and to develop vaccines. But I think going forward, these kinds of problems could be solved even more quickly if we were all open with our observations and our publications and collaborative. Now, one feature of the coronavirus epidemic that has actually been good for science is that all of the scientific journals that collect literature, publications on the coronavirus, decided to allow the work that's published to be made available without a subscription. So even the very high-profile journals that are expensive, that libraries have to subscribe to, have allowed all of the research on coronavirus to be made available so that everyone, irrespective of their location, even in the public, is made aware of the recent observations. Furthermore, many of these publications have, even before they've been published, been posted on an archive like the bioRxiv or the medRxiv, which is only a copy of something that existed in the physics community, the physics archive, almost 40 years ago. And in these archives, every manuscript is posted when it's about to be submitted for publication and is available for everyone around the world to see. And this has had enormous benefits because we've been able to learn information as soon as it's going to be made available. And we've been able to criticize these publications even before they're published. Some have been taken down because so many people have an opportunity to review the data that there's a chance to make critical comment. Now, the coronavirus also serves as an opportunity for me to tell you just a little bit about the kind of cell biology that I do in my lab and that Liwen does in his lab, and that has to do with biological membranes. And I illustrate this in this slide again by pointing out that the virus is actually a membrane. It has a lipid bilayer membrane and proteins on the surface. An are, for instance, the spike proteins that are the target for immune attack. And the lipid bilayer surrounds an RNA genome core inside of what is otherwise a very large membrane, round membrane. Now let's talk a little bit about membranes. I know this is a broad audience. Not all of you are cell biologists, so let me very briefly tell you a little bit about what a membrane is. This is a cartoon of a biological membrane. It consists of two layers or leaflets of lipid molecules, phospholipids, where the surface exposed to the outside of the cell or to the inside of the cell is occupied by a head group that is very often charged and polar, water-loving, hydrophilic. Interior, bound to these head groups, are hydrocarbon fatty acid side chains that provide the interior with a very apolar hydrophobic environment. So these bilayers are by themselves insulating. They don't allow anything back and forth very efficiently. And so membranes have to be populated by channels or other proteins, such as these green structures, proteins that may emit selective ions or small molecules, or other proteins that may represent surface receptors that, for instance, bind hormones. So all of the membranes in a cell have a personality that is dictated by the combination of these lipid molecules and protein molecules. Now, here's another kind of a cartoon of the two major types of cells in the world. This is a eukaryotic cell, this larger structure, and in the upper right-hand corner is a bacterial cell, a prokaryotic cell, much simpler. Our cells, or unicellular eukaryotes, are more complicated. the chromosomes are organized in a nucleus that is bounded by an envelope of two membranes. And the cytoplasm, where much of the activity that allows a cell to grow and divide occurs, is also bounded by many different organelles, structures such as these strands, which are involved in biosynthesis of proteins that are going to be secreted from the cell. Or a structure such as this, the lysosome, which is a kind of a digestive organelle where proteins are sent to be degraded. Or the powerhouse of the cell, a mitochondrion that is producing the energy currency of the cell called ATP. Bacteria are much simpler. They have much of the same metabolism, but it is not as highly organized. Now, just to summarize a great deal of work in the middle part of the 20th century, we now know that proteins such as the hormone insulin have to be organized in their export from a cell. The human genome encodes something like 23,000 different protein molecules. About 30% of those protein molecules, although they're made inside of the cell, have to be shipped outside of the cell, exported by a process called secretion. They have to have special signals on them that allow them to gain access to this network of membranes, shown here in this kind of salmon color, starting with a membrane that's bounded on the outside of the nucleus, going through another elaborate network called the Golgi apparatus, and finally into granules that are discharged at the cell surface by a process called membrane fusion. This is an enormously complex pathway that was first elucidated using techniques like microscopy by a very pioneering cell biologist by the name of George Palade. In my laboratory, we investigated this process in a simple organism, baker's yeast, and were able to discover the genes that organize that process. Now, I say this all because it actually relates to coronavirus, because coronavirus has a membrane around it, and it uses this pathway. It uses these membranes for its assembly in an infected cell. And I'll show you a couple of images of how that works. This is now just a cartoon. Here's the virus as it's about to invade a cell. It exposes on its exterior, on the surface of its membrane, these proteins that you've heard of called the spike protein. The spike protein has a special target on a receptive host cell that happens to be another protein that has a different purpose, but the virus has evolved to exploit this protein as its entry portal. The virus then binds to this receptor and is swallowed up into the cell that it's about to infect by a process called endocytosis. The virus gets swallowed up, and then the virus membrane has to fuse with a host membrane inside of the cell for the RNA genome of the virus to be ejected into the cytoplasm to make new virus particles. And here is a section, an electron micrograph thin slice through a coronavirus infected cell, showing the enormous involvement of intracellular membranes in the infection and in the biogenesis process. New virus particles are produced in the cell on membranes, and they are ejected by a budding process that starts at the cell surface. So a cell can produce many virus particles without itself necessarily being killed. And so the cell can continue to manufacture viruses, and they thus can invade very rapidly. And we've learned an enormous amount about this virus through the international effort that has gone along to try to understand the virus and to develop new therapies. Now, I want to turn to a different topic, but I think it's related, and it's related to the question of publication. I told you that during the pandemic, we've had the really unusual opportunity to be able to see all of the literature related to coronavirus without having to pay the subscription fee, at least for those journals. Now, scientists, many of you in the audience, are keen to publish in certain selected journals that are so-called high-profile journals. And these journals are very selective, and they often cause an investigator to spend a great deal of time and trouble in preparing the paper for publication. But that has some unfortunate consequences that I would like to discuss very briefly in this section. So this is relating to the challenge in the communication of scientific research. Now, there's a vast literature in the life sciences. Probably several million publications are published each year in thousands of different journals. But as I said, many investigators prefer just a small number of journals that are highly selective. And the process of review of these highly selective papers has some flaws that result in the publication of results that cannot be reproduced. In fact, alarmingly, some years ago, pharmaceutical industry companies investigated whether key observations in an area of cancer biology could be reproduced under the controlled environment of a commercial company where things have to work to be useful. And so here is a paper that was published in one of these competitive journals, Nature, that was conducted by a team of investigators at a very large biotech company in California called Amgen. The principal investigator is an Australian scientist by the name of Glenn Begley. He and his team identified a number of very prominent papers in cancer biology, and they sought to try to replicate certain key observations in these papers. These are papers that they at Amgen would have used to try to develop targets for chemotherapeutic approaches. So they were really keen to see if these observations were worth their investment in an effort that might cost $1 billion to produce a drug. And the shocking discovery that they made in this replication effort was that of 53 prominent papers, only 6 had key experiments that they could reproduce in the laboratory, namely only 11%. The vast majority of these observations in these papers could not be reproduced. So that's alarming. It says an awful lot about the scientific enterprise. And of course, the governmental funding agencies that in the United States spend tens of billions of dollars on biomedical science were and are very concerned about this. They wonder, what is their investment going into? Fraud? Is this something that is fundamentally flawed in the way scientists do their work or have their work reviewed? Now, after this, this publication, others reported similar observations. There were various explanations, but unfortunately in this paper by Begley and Amgen, they did not publish themselves the exact experiments that they attempted and which could not be reproduced. They just didn't have the time and trouble to make a publication out of that. So at that time, I was the editor-in-chief of a new life science journal called eLife, which has an open access policy and an openness in the way that we review papers. We were approached at eLife by an organization called the Center for Open Science in the United States. And they had identified another 50 papers in cancer biology, and they raised funds and they identified laboratories, contract laboratories that were willing to attempt to reproduce some of the observations. And more importantly than in this example from Amgen, they wanted us to cooperate with these contract laboratories to publish their findings in 2 forms. The first was a paper called a registered report, where the contract laboratory would simply write down in great detail exactly what they were going to do to attempt to replicate the published experiment. We then received these manuscripts, we reviewed them, we had our editorial team examine them and outside experts. And if we felt that they really were doing an adequate job in attempting to replicate these results, we accepted the manuscript, and that was published as a registered report. Those registered reports then were followed by a bona fide attempt to replicate. And this is now several years old, but the project, which was very time consuming, very labor intensive, was called the Reproducibility Project in Cancer Biology. In total, there were 37 publications in the most selective journals that we commissioned for repetition, but the results in the end were rather difficult to interpret. Let me just at this point tell you a bit more about how we review papers at eLife. At eLife, when a paper comes in this case it was true of these papers as well, the paper is considered by a member, actually several members of the editorial board, and if it's considered appropriate in the subject areas and at a level of excellence that we expect, we then commission reviews from scientists around the world. These scientists then render their opinion independently, but unlike other journals, the scientists come together in an online forum to compare notes and to decide which experiments would necessarily be needed to be improved, or whether the paper just wasn't going to pass muster. And as a result of that consultative effort, a decision letter is written by the member of the editorial board, and that's sent to the author. So that's a different way of handling papers that we feel is more open, because the reviewers, when they serve in this collaborative team, have to be identified to each other. So as a reviewer, you can't hide your identity, at least from the other referees. Well, so we, as I said, we conducted this study in two forms: a registered report, which is simply a statement of what the contract laboratory was going to do to replicate. These were peer-reviewed, and if they were found appropriate, they were published as registered reports. The experiments were then conducted by the contract laboratories. Once again, they wrote up the papers. Their results were then sent back to the editorial board and to outside experts. And there was often a back and forth with difficult experiments. And in the end, almost all of them were published as replication studies. The outcome, just to summarize an article that was published in Science, was mixed. Several, in the first 5 of these replication studies, 2 of the key results could be reproduced in 2 of the papers. And in 2 others, they could not be reproduced. But in a 5th, it just wasn't possible to set up the experiment because the reagents weren't the same, the antibodies weren't the same. There was some change over time that made it difficult to replicate the study. So our conclusion from this, after a great deal of work, is that replicating research in this kind of very formalized way is expensive and time-consuming, and in the end, doesn't necessarily give you the results that would either give you comfort or not in the conclusions of the papers. The best way going forward is the good old-fashioned way, and that is if a result that's published is important, it will be reproduced in other laboratories. If not, it will disappear. Unfortunately, sometimes fraudulent research is published, and that can do a great deal of damage. So the review process is a critical component of the nature of the publishing enterprise. And I'd like to turn to that issue right now and to discuss with you my personal view, and I think it's shared by many, about problems with the peer review system itself. I think, that some of the problems associated with the publication of results that are irreproducible has to do with problems in the way that papers are reviewed. Now, in the most selective journals like Science and Nature, the decisions about whether a paper is going to be reviewed or ultimately published are made by professional editors, not by active scholars. Now, let me describe these professional editors. They are all trained scientists. They all have PhDs. Most or many have done postdoctoral work, but that work may be many years ago. And when they come to a broadly based journal like Nature or Science, they are often called on to evaluate papers that are well outside of their area of expertise. In fact, it's quite usual that a young scholar, fresh from a postdoc, will go in and suddenly have to deal with papers with which he or she is completely unfamiliar. They then have to do background reading, they have to find the appropriate experts to consult, but then in the end, they are called on to make a final decision, more or less unilaterally by themselves. And this, I think, is a real problem because many of these people, though well-intentioned, and having been trained in the past, don't have the appropriate judgment to decide which of the comments of the referees, which are often very different, which are most important for the author to complete. So more often than not, for those of you who experience this, at one of these journals run by professional editors, the paper comes back and you're asked to just essentially do everything that the referees have found wrong. This puts an enormous burden and pressure on the students and postdoctoral fellows in the laboratory to make sure that the paper will eventually pass muster and be accepted by the editor, the professional editor. Now, unfortunately, I feel that puts an undue burden, an undue pressure on young scholars. And I'm afraid all too often, corners are cut and data is cherry-picked. And sometimes wrong results get published with the best of intentions just to make sure that the paper is published in a prominent journal, because many people feel that if they don't publish in these journals, they won't have a successful career. I think this is a flaw in the system and a pressure that I would really love to see released in some way. Now, I'm not the only one who feels that the system is broken. Two very prominent scientists who themselves have published in these journals have made comment on this. Hiddet Pluch, an immunologist at MIT, publishing an editorial in Nature, said, peer review of scientific papers in top journals is bogged down by unnecessary demands for extra lab work. This is what I told you about a moment ago. Often investigators will cut these things short just to get the paper published. Or Martin Raff, Sandy Johnson, Peter Walter, Johnson and Walter at UCSF, publishing in Science, the stress associated with publishing experimental results can drain much of the joy of practicing science. It was for this reason that we decided to start eLife, to try to have all of those decisions made by active scholars, experts in a particular area, and to confer with each other to throw out those comments that may not be necessarily pertinent to the study. Now, another very bad influence that has put enormous pressure on journals and on scientists is the use of a number that is, I'm afraid, all too often considered by young scholars, a number called the journal impact factor. This is a number that was created many decades ago by an organization called the Institute for Scientific Information. It is basically a crude measure of the number of citations that a journal generates per article over a 2-year window of time. And there are many bad consequences of the use of this number, not the least of which is its inaccuracy. Such a number could not possibly be computed to this level of accuracy. And journals used to promote, and many still do, promote this number shamelessly. Nature used to prominently feature this in their advertising. And that would, of course, attract more papers, because people think if it's got a big impact factor, that means that my paper is going to get more attention. Unfortunately, I think that the consequences of this are pathological. In fact, subsequent to their use of this number, a number of us were able to persuade Nature to do away with this, and the editor-in-chief of Nature at the time published a very important editorial calling for the remodeling of this journal impact factor. And he said that Nature and Nature journals were going to diversify the sorts of information they used. And he says in his editorial here, metrics are intrinsically reductive and as such can be dangerous. Relying on them as a yardstick of performance rather than as a pointer to underlying achievements and challenges usually leads to pathological behavior, just what I said. The journal impact factor is just such a metric. Now, unfortunately, in spite of this editorial, and in spite of lots of us complaining about it, many journals still prominently advertise this number in an attempt to generate publicity for themselves. And the consequence of that can be felt around the world. And I use as an example, one that you people felt in China through a bulletin that was published by the Chinese Academy of Sciences some years ago, which basically offers a cash reward, the equivalent in the US of $33,000 US back then, for anyone who happens to win the lottery and to publish in a journal like Cell, Nature, and Science, with less money being offered for other journals and a considerably lesser amount being offered for domestic journals. I understand that scholars in China who are very successful in this enterprise would often make a substantial additional sum of money for their salary just because they happen to publish in Cell, Nature, and Science. And I'm telling you, this is not necessarily a measure of scholarly achievement. It's simply a measure of finding the right editor and finding the right time and day and subject matter to interest his or her, attract his or her attention. Well, I hope I've persuaded you that I certainly feel very strongly, and many of us share this view, that the impact factor is not an appropriate way to measure scholarship. And so a group of journal editors assembling some years ago in San Francisco at the meeting of the American Society for Cell Biology created a document called DORA, the Declaration of Research Assessment, where we basically call on all the clients of journals, or the publishers, the editors, the scientists, and the readers to move away from the use of this basically meaningless number called the impact factor and to assess outputs on their own merits. It used to be that people would assess the quality of science by reading the paper rather than simply relying on a number. And there are other tools and approaches that may be used, but the best is to actually read the paper to decide whether it has merit. This declaration has now evolved. There's an organization that sponsors this. There are many more than tens of thousands of signatories around the world who share this. But I have to say, unfortunately, many people who sign this document nonetheless succumb to the pressure of this meaningless number and publish in journals that just because they have that impact. Now, many institutions have promoted another very important aspect of scientific sharing, which is open access. And so I'm sure you all know about this. Open access is the model where you as a scientist pay the fees associated with the publication of your own work. And it's then open for anyone to see, irrespective of their academic or physical location, even the public or physicians or small biotech companies that can't afford the license to have these journals can read the fruits of labor. And it only makes sense that journals and scientific papers should be openly accessible. It is, after all, the governments that fund most of the research, most of the basic research that's conducted around the world. And the citizens of those countries deserve the opportunity to see the fruits of their labor, to see the work that's published at their expense. And yet, citizens in various countries have reported to me that if they saw some interesting article referred to in a newspaper and they went to the journal site to read the paper, they had to pay an additional fee of $30 or more for the privilege of seeing a paper that was funded by their tax dollars. So this is wrong, and many of us have supported what is called the open access movement. Most dramatically, in the past few years, my institution, indeed the entire University of California, which accounts for 10% of the scholarly output of the entire United States, had an unsuccessful negotiation with the largest publisher of scientific literature, the journal Elsevier, and they decided after 2 years of fruitless effort to suspend the contract to Elsevier until Elsevier was willing to change their publication policy to allow all of the work of University of California scholars to be open access when it's published in the Elsevier journals. Journals like Cell or Lancet and all of the clones of those journals are owned and operated by Elsevier. Elsevier is a very large, very profitable organization. Their profit margin exceeds that of any of the Fortune 500 companies that have much bigger volumes of business. And of course, they have a perfect business model. They charge you to publish your paper there. They charge you through your institution to read the paper that you've published. And they charge the public who paid the tax dollars to do the research to read their papers. And on top of it, they get a lot of free labor. All of the scholars who are asked to review papers for these journals do so for free. Their professional editors, of course, are paid, but the scholars who actually do the heavy lifting and read the papers do so for free because they consider it a scholarly obligation. As a result of that, the business model for the most profitable commercial enterprises like Elsevier and Nature are enormous. They have an enormous excess profit, and they're quite aggressive in seeking contracts with institutions, indeed with whole countries. University of California fought this, and at one point I was asked to participate in a radio interview, and I found a very interesting number that was, I thought, quite simple and very illuminating, that expresses the difference in the profit that Elsevier makes on their publications compared to a journal that's published by a scientific society that calls for no profit, that in fact mandates no profit for their journal. So I'll compare the profit that Elsevier makes compared to the cost to the university of an Elsevier journal and the cost to the University of California of a publication in the journal, the PNAS, the Proceedings of the National Academy. Elsevier charges the University of California for its contract to the bundle of journals that it makes available, approximately $11 million a year. So, but how do you evaluate that number in a budget to the University of California that's in the billions of dollars? Well, there is a way, because you can compare that to the contract cost that the University of California pays to the National Academy of Sciences for the privilege of having access, online access to the articles published in the PNAS. If you compute the cost to the university for every download of a PNAS paper, the cost is approximately 4 cents, 4 or 5 cents per download. The cost per download of a paper published in the bundle of Elsevier journals, the cost to the University of California was over $1 per download, approximately 20 to 25-fold difference in the profit margin that Elsevier enjoys. Well, it was for this reason that the university insisted in its negotiations that this had to change, and Elsevier had to bend and allow journals to be made, journals to have papers published by our scholars to be openly accessible. Well, I can report to you now that this year we have finally successfully concluded a negotiation with Elsevier, and they have agreed to the terms of the University of California contract so that papers published by UC scholars will now be open access. Elsevier will still make money, but perhaps not quite as much. Now, let me conclude this part of my talk with some suggestions that I've made on various occasions that journals should embrace to try to make the work more reproducible and more openly accessible. One of the problems with many of the journals, particularly the top-ranked journals, is that when publications are found to be wrong, that conclusions are found to be wrong for whatever reason, they are very reluctant to allow an investigator with claims that oppose something that they've published to publish that work in that journal. They are very resistant to challenging work that they decided was appropriate. It's very difficult to get a journal like Nature or Cell to retract a paper that they find had mistakes. I think journals ought to own their own mistakes. They ought to be willing to provide more space for the publication of results that challenge work that is published in that journal. I think it would be a gesture to openness, and it would show to the authors that they mean business, and that if there are real challenges, that they will be revealed. The review process should be more open. I think what we've done at eLife is an example of the kind of more open review process that would help, where the reviewers confer with one another, either knowing their names or even anonymously. At least they would share their impressions of a paper and come to some synthesis about the review. And then in addition, I think it's important that the reviews themselves be published along with the final paper. A number of journals do this. Now, when there is a charge of manipulation of data, and this is the most serious charge, where a mistake is made, conclusions are wrong, but there is evidence of manipulation, of fraudulent manipulation of the data. The problem is how to investigate such a claim. The journals are not investigative bodies. They don't have the wherewithal to investigate detailed claims of fraud or manipulation. So very often they proceed by turning the investigation back to the home institution of the author of the article. And some institutions will take that seriously, but unfortunately other institutions are more interested in protecting the investigator who's been charged with manipulation, and they don't conduct an honest investigation. So very often these things get, as we say, swept under the rug. I think there should be a change in the procedures of investigation that require the home institution to accept advice and guidance and counsel from some outside body in how to proceed. Item number 4, we're now realizing this, as I indicated earlier. Many people now are encouraged to and actively embrace the idea of publishing the preprint of their paper. Whenever we are ready to publish a paper in my laboratory, many laboratories, we send the paper to the bioarchive for posting, and then we send it into the journal to begin the review process. The version of the paper that's eventually published will change, and new versions of that can be posted on an archive. I think there's an enormous benefit to have even more eyes, more people see the work and be able to comment on it, to find flaws in it that may allow the investigator to improve the work. And finally, as I've already emphasized, I think it's critical that we disavow this meaningless number, the journal impact factor, because it only creates more difficulty for everyone involved. And I call on all to embrace the principles of the Declaration of Research Assessment. Now, I'd like to conclude the last part of my talk with an example, I think, of a very new and I hope important collaborative effort that I have undertaken at the behest of a very wealthy philanthropic donor in the United States, in collaboration with an organization called the Michael J. Fox Foundation. Now, for personal reasons, I have a vested interest in Parkinson's disease. I have followed it over many years because my wife suffered from the disease and eventually died of Parkinson's a few years ago. After her death, I was asked by a representative of this philanthropist if I would consider helping to organize a panel of neuroscientists and Parkinson's experts to consider the key areas of basic science that could use a large infusion of capital to help understand the origins of the disease. The benefactor himself has a family history of Parkinson's, and he came to the conclusion quite independently that with a disease that we've known for now 200 years, the progress in developing a real effective cure rather than just a palliative treatment has been quite limited. And he came to the conclusion, one that of course many of us support, that the problem is that we just don't have a basic understanding of the molecules and the cells that go bad and when they go bad and what happens at the onset of this disease. How is it that the disease comes about? So I, of course, was really quite happy to turn my personal grief into something constructive. And so I agreed to become what is now an organization called Aligning Science Across Parkinson's Disease. And let me just tell you a little bit about where we come from. So many diseases have seen tremendous progress in recent decades. I mean, cancer and heart disease. There are solid discoveries. There are molecules that allow us to follow the progress of these diseases. And there are now increasingly effective treatments that are saving thousands, indeed millions of lives. And yet we are experiencing around the world a pandemic of a different sort, one for which there is no vaccine, a pandemic of neurodegenerative disease, principally Alzheimer's disease and Parkinson's disease. This is a pervasive problem. It's around the world, and it is only growing in complexity. And we only have a kind of a foggy picture of the puzzle of this disease. So let me tell you just a little bit more about what we know about the disease. It is a pandemic in the real sense. The incidence of the disease of Parkinson's is rising quite dramatically. In fact, even more dramatically than Alzheimer's disease. It's likely in the next more than a decade to afflict almost 20 million people. It knows no boundaries, just like coronavirus. The prediction is that in China, over half of the new cases of Parkinson's disease will be detected, will be diagnosed within the next decade. And so this is a concern to all of us. And I feel, and many of my colleagues feel, that the only way we can tackle a complicated problem like this is to collaborate. This is the kind of a challenge that I feel is beyond the capability of an individual or even a talented laboratory to tackle. We need a collaborative effort. Well, let me tell you a little bit about what we know about the disease. It's been known now for some decades that one of the deficits as a patient progresses is the loss of a subset of cells in the midbrain called the substantia nigra. These are cells, dopaminergic neurons, that make the neurotransmitter dopamine. This can actually be seen. These cells can be seen because they also produce a pigment called melanin. And here's a section of a brain of someone who died for other reasons. One can see these melanin-containing cells, which actually are coincident with dopaminergic neurons. Now, when a patient first presents with the symptoms of Parkinson's disease, they have already lost, it's estimated, half or more of the dopaminergic neurons. And by the time they die of the disease, here's a section of the brain of someone who died of Parkinson's. These melanin-containing pigmented cells are virtually gone. And of course, one of the treatments for Parkinson's is the use of a replacement form of the precursor of dopamine, L-DOPA, which can be administered and has some palliative effect, but it's not a cure. And the effect of that drug wears off after a few years as the cells that are making dopamine in the natural physiologic setting increasingly die. Now, 100 years ago, a British physician sectioning the brain of someone who died from Parkinson's disease, a fellow named Louis, detected in the substantia nigra a population of cells that had not yet died, but had accumulated structures of what turn out to be aggregates of protein, including a protein called alpha-synuclein, a neuronal protein that was first discovered bound on the surface of the little vesicles that carry neurotransmitter. But this protein tends to aggregate, and there are, in fact, mutant forms of this protein that aggregate even more efficiently. And patients who have a mutation in the alpha-synuclein gene often succumb to Parkinson's at an earlier age because of the more pronounced accumulation of these structures. Well, these Lewy bodies, unfortunately, can only be seen, can only be detected, visualized in the biopsy of a patient who's died. It has not yet been possible to visualize these structures in a living person. And that's really a serious problem. It would be wonderful just for diagnostic purposes and for following the progress of the disease if we were able to visualize this by some non-invasive dye. And that people are working very hard on that, but it's not yet available. Now, in the progression of Parkinson's, it's imagined through reconstruction of patients who've died that these Lewy bodies may originate in early stages at the brain stem and progress elsewhere in the brain, including to the substantia nigra, but also to regions of the cortex. Indeed, about 30% of Parkinson patients suffer from dementia, and there is evidence that is at least correlates with the spread of Lewy bodies throughout the brain in the very end stage where there are serious emotional and cognitive disturbances that precede death. Now, we can see these Lewy bodies in cultured nerve cells. They can even be reproduced in the laboratory. Here's a paper that was published recently from a laboratory in Switzerland showing in a false color image a thin slice through a nerve cell which has been induced to accumulate alpha-synuclein in a fibrillar form. And it shows that this structure, the Lewy body, is rather more complicated. It includes not just protein aggregates, but also fibrillar proteins and membranes and other sorts of particles. And this is an immunofluorescence image of the Lewy body that can be recapitulated in these nerve cells. Now, I have to caution that although these Lewy bodies are associated with pathology, it has not yet been proven that the Lewy bodies are the reason that dopaminergic neurons die progressively as the disease advances. They may be a byproduct of the illness, not a cause of the illness. So that's how ignorant we are about the basics of this this disease. Now, so I told you about one gene, alpha-synuclein. Turns out through genetic studies of families with Parkinson's disease throughout the world, laboratories have discovered over 20 different genes, mutant alleles of which predispose to a genetic form of Parkinson's. It's estimated that these genes may account for as much as 30% of all of the cases of Parkinson's. That leaves, of course, a very large fraction of the disease unexplained. That may yet be revealed as a genetic form of the disease, or it may be some as yet unknown environmental influence that causes the disease. Let me, in the brief time that I have left, tell you about three other genes actually, that are known in families, certain families with Parkinson's disease, that has some very interesting effects on cells that can be explained, or that could be used to explain why dopaminergic neurons die when patients have these mutations. Now, many of you who've had a biology class will know about this structure. It's called the mitochondrion. It's the powerhouse of a eukaryotic cell. Our cells, cells that have a nucleus, also have a mitochondrion. It's organized in a rather complex manner with an envelope that surrounds a very protein-rich interior and membrane infoldings that are busy, actively engaged in oxidative metabolism and generate a large fraction of the ATP that cells require as the energy currency to make things work. Now, the mitochondrion is a very sophisticated organelle. It even has its own chromosome, a small genome, the mitochondrial genome, that is housed in the cytoplasm called the matrix of the mitochondrion. And within these infoldings, these membranes called cristae, there's a very organized transfer of electrons, finally, to the final product, which is water. But along the way, an oxygen-free radical is generated at the very end of the electron transport chain. And that free radical is held by a complex, a cytochrome oxidase protein complex in this membrane that coordinates through metal ions this free radical oxygen and holds onto it, protects it for its eventual destination to form water. Now, if the mitochondrion is damaged for whatever reason, and just the normal course of cells growing and dividing, some mitochondria become damaged. There are two bad things that happen when a mitochondrion becomes damaged. One is the genome, the DNA of the mitochondrion may escape into the cytoplasm of the cell. Cells don't like DNA in the cytoplasm. They like DNA to be kept in its place. And if DNA happens to be leaked into the cytoplasm of a eukaryotic cell, the cell thinks something has gone wrong, and it sets in train a pathway that ultimately leads to the death of that cell, a stress pathway that commits the cell to death so as to prevent further damage to surrounding tissues. Another thing that happens that can be bad about a mitochondria being damaged is those oxygen-free radicals, if they happen to be released and can diffuse into the nucleus, they will cause DNA damage. They can break double-stranded DNA. So you want to protect against these two releases. And so cells have, it turns out, evolved a quite elaborate scheme that is a surveillance mechanism that identifies damaged mitochondria and puts a little tag on it. It's a tag that says, destroy this organelle before the cell has to destroy itself. The way the cell does this is by a process called autophagy, or a specialized process called mitophagy. where the damaged mitochondrion, in this case with a tag on it that I'll tell you about in a moment, the damaged mitochondrion is enveloped by a membrane in the cytoplasm of the cell, delivered to the lysosome, which is the digestive tract of a cell, and many different enzymes in the lysosome, hydrolytic enzymes, chew up the mitochondrion and thus relieve the cell of the potential of releasing damaged material into the cytoplasm. So this is a very important pathway of autophagy, or specialized for mitochondrial destruction, called mitophagy. Now let me bring this back to the very interesting mechanism that cells have evolved to tag damaged mitochondria. This can be done experimentally with human cells in culture. If you add a drug that causes the membrane potential in the inner membrane of the mitochondrion to be dissipated, the cell recognizes the mitochondrion as malfunctional. And what it does is it uses two different genes, the products of two different genes called PINK1 and parkin, to put a special tag on the outer surface of the mitochondrion. This is a tag that's made of a little short piece of a protein, a peptide called ubiquitin. And this ubiquitin tag is only added to damaged mitochondria. And that signal, the ubiquitin signal, is recognized by the capture of these damaged mitochondria by an envelope membrane that swallows up the damaged mitochondria to deliver it to the lysosome. Now, the important thing, and this is really critical for Parkinson's disease, is that these two genes, PINK1 and parkin, were discovered as genetic forms of Parkinson's. Families with mutations in either of these genes fail to tag damaged mitochondria. They thus persist in a cell. And just as I indicated earlier, the persistence of a damaged mitochondria will lead eventually to the cell killing itself to save surrounding tissue. And the dopaminergic neuron is particularly stressed in such circumstances, and thus it dies prematurely. Now, finally, let me tell you about another gene that was part of the 20 genes that were identified in patients with families with Parkinson's disease. It's a gene called LRRK2, LRRK2. It's codes for an enzyme that adds a different kind of a tag to other proteins, a phosphate tag. So this is a protein that many of you will know of as a protein kinase. It's an enzyme that takes ATP and transfers one of the phosphates from ATP onto a target, and that often changes the target to do something else. Now, a group in Scotland and now a colleague at Stanford have collaborated to develop a very interesting story that I don't have enough time to tell you about, that explains why this protein, LRRK2, may cause the selective loss of dopaminergic neurons. It turns out that this protein, this kinase, LRRK2, when it's mutant, and only when it's mutant, is hyperactive as a kinase. It is much more active in attacking and attaching a phosphate to its target. And when that happens, cells that have a sensory structure called a sensory cilium fail to make this structure. The cilium fails to be made when the target of LRRK2 is hyperphosphorylated. And that causes a loss of communication between this cell in the striatum of the brain, a cholinergic neuron, loss of communication between this cell and dopamine neurons. Under normal circumstances, dopamine neurons are under stress, and that stress is communicated to these cholinergic neurons through this cilium, which is turned on to allow the production of a growth factor that sustains dopamine neurons. So this is a vital connection between these two cells, a functional connection. In patients with a mutation in the LRRK2 gene, this stress cannot be recognized by the cholinergic neurons because they don't have the sensory, the antenna that detects this signal. And as a result, they don't bother to produce this growth factor. And thus the dopaminergic neurons are no longer nourished and they die. So this is another very different aspect of what may be going on, at least in a subset of Parkinson's patients. Well, let me conclude now by just very briefly telling you what we're trying to do about this. I mean, I've given you 3 very different snapshots of what this disease entails, at least at the genetic level, and it's almost certainly going to be more complicated. It's a puzzle. It's a 5,000-piece puzzle, and what we need to do is put the pieces of the puzzle together. So over the past 2 years, a group of us has developed a plan through this organization, Aligning Science Across Parkinson's. And what we've done is to decide that we want to promote an international, completely international collaborative effort involving laboratories around the world who are organized in teams of 4 to 5 principal investigators in their laboratories, teams that were picked because they already had been engaged in some kind of collaboration. So they know what it means to have the give and take of a collaborative effort. These teams then were selected because they had Parkinson's experts or people from other fields, and they put together very interesting proposals. And just last year, we identified the first 21 teams in this effort. Let me tell you what we've looked at specifically in terms of the research activities we wish to promote. The first pillar of our effort is to understand the complete genetics, all of these genes, their proteins, the pathways that they are engaged in, how these different genes and their proteins may interact with one another. Another pillar of our effort is the immune system. There's some evidence that the brain lesions, these Lewy bodies, and even with Alzheimer's disease, the plaques that build up in patients may be inflamed by a neuroinflammatory process or by the immune system outside of the brain. What is that connection? What is the inflammatory process that may make the disease worse or perhaps even initiate the disease? At an even more fundamental level, we don't understand all of the connections that dopaminergic neurons make. I told you about one to a cholinergic neuron in the striatum, but there are many other inhibitory and activating interactions, and we don't fully understand that circuitry in the human brain. And finally, and perhaps most mysteriously, there is a prodromal phase, often decades in length, preceding the onset of the movement disorder. And there are certain common symptoms that emerge in patients who eventually present with the disease, who reflect back on these common symptoms. One of the most interesting and unfortunate symptoms that progresses with very high frequency to Parkinson's is an illness called REM sleep disorder. And this is an illness where people are unable to distinguish dreams from actual wakeful thoughts. And when they have nightmares, they act out their dreams while they're sleeping, often flailing around in bed, harming themselves and harming their partner. It's a mysterious syndrome, but something like 80% of people who present with that syndrome eventually, some years later, succumb or then present with the movement disorder of Parkinson's. We'd love to know what the connection is between those syndromes and the eventual illness. Indeed, we'd love to have biomarkers, biological molecules that we could follow that would be diagnostic of disease progression. No such molecular marker yet exists. And finally, let me just tell you why this ties into the theme of my presentation. We at the ASAP program feel very strongly about open science and how it facilitates the rapid and free exchange of ideas. And thus, all of our investigators, we now have 100 different laboratories, and we're now looking at proposals from even more. All of our investigators must commit to being open within this network, to sharing new ideas and results, and they must post all of their work when it's ready for publication on a preprint archive, and they must agree to publish their work in gold standard open access journals, that is, journals where all of the papers are openly accessible. We feel strongly about this because we think this problem, indeed many other problems in science, will best be tackled on a collaborative, with a collaborative approach that builds on and reuses and openly criticizes the published body of scientific knowledge. For science to effectively function and for society to reap the full benefits from scientific endeavors, It is crucial that science data be made open and not hidden behind a firewall or hidden in a laboratory. The ultimate value of data cannot be predicted, and thus with advancing technologies and analytics, data that's already been published can be reanalyzed when it is in an open access format. So I'd like to thank you for your attention, and I'm pleased to be able to share these thoughts with you, and I'd now be happy to take questions if there are any.
All right, thank you again, Randy, for excellent, also inspiring talks. Okay, so right now we have time for questions. Okay, we can open question to the floor here, also to the audience in the Zoom. Okay, for the Zoom audience, they can just unmute themselves, ask the question, right?
You can type the question in the chat.
Or you can type your question in the chat box. Okay. All right. Any questions for Randy? All right, Chunshan.
Professor SCHEKMAN, thank you so much for a very insightful and then also exciting lecture. I actually also thought about a few questions at the end. We often actually see the drive to publish papers in so-called Nature or Nature series versus the professional journals, and that actually publish the professional area. On the other hand, Nature keep producing this Nature this, Nature that. So how do you say that, and what's your advice to our young researchers, young faculty members in that area?
Yeah, no, this is a pervasive problem. It's not just at your institution; it's worldwide. Nature and Cell and Lancet and these so-called high-impact journals they're very good business people behind them. They have branded themselves very effectively, and they have captured a market that is difficult to shake. Indeed, the attitude that many of your colleagues feel, and mine here as well, is that they have to publish in these journals. They have no choice. If they don't publish in these journals, their work won't be recognized. And it's a bad incentive system. So I won't publish in these journals anymore. And some of my colleagues won't publish in these journals anymore. But of course, you can say, well, of course, I don't have to publish in these journals. What do I tell a young person? Well, what I tell a young person is, turns out it is not essential to advance your career. There are people who have never published in these journals who have done very well. And secondly, as a young scholar, the most important thing for you is to get your work published soon, rather than waiting the sometimes years that it takes to get a paper published in one of these fancy journals. I once met a Mexican scientist who told me proudly that he finally published his paper in Cell. It only took him 4 years to publish it. Now, what a mistake that is. He should have published it in a journal that had reasonable standards, where the decisions were made by his peers and not by professional editors. Now, I know that I've said this many times in many different forums, and I know people will be skeptical, but there are, in my policy in my laboratory is that I'm happy to publish anywhere where the decisions are made by active scholars, where all the decisions are made by active scholars. These are the journals that are run by the professional scientific societies, and many of them are outstanding journals, the PNAS, the JBC, the Journal of Cell Biology. I mean, in my field, there are many fine, and eLife, of course, there are many fine journals, the PLOS journals, there are many fine journals. And if we all just got away from this obsession with the branding, the brand name, we would all be in a better place.
Thank you very much. I have another question related to the idea of publishing or open posting before publication. I was serving as the chair of 2 different divisions in the American Chemical Society. We used to actually publish the preprint before we publish formal journals. And then American Chemical Society journal editors strongly against the idea. They actually say, if you publish a preprint, we will not allow you to publish in journals. Are you aware of this trend?
Oh yeah. Well, I have to tell you, the American Chemical Society is just as bad as Elsevier in Nature. They are profit-driven organization, and they have been very vigorously opposing efforts at open access. So I don't respect that organization for that reason. There is no reason whatsoever to not post your paper on a preprint archive. And if a journal tells you, I don't think many of them do this anymore, but if they tell you that they're not going to consider your work, go elsewhere.
Thank you for your comment, and that's very encouraging. I fully agree with you.
All right. Question from the audience. I think from the Zoom, you can just raise your hand and we will unmute you, and then you can ask the question to Randy directly.
Okay, here's a question from the chat. Edwin Chan asks, would you please advise how neurobiologists at CUHK can join the ASAP and participate in its research activities? Well,
right.
We advertised around the world for teams of investigators to apply. There were no national boundaries whatsoever. And unfortunately, the vast majority of the applicants that we got were from Western countries. We have only one Chinese scientist who has applied in this most recent round, which really to me is very disappointing. So I'd like to ask Edwin Chan if he was even aware of the existence of this program. We published in eLife, we published an article describing the initiative before we called for applications, and scientific societies around the world were alerted to this, to the availability of funds. And I should tell you, we have already committed almost $350 million to these teams around the world. So we have a very generous donation from our benefactor. There will be future calls for proposals. Just keep your ears open. But right now, for now, we've committed. The network is now getting to the size where we don't want to make it much bigger.
Right. Next questions. Okay, Yilin here.
Thank you, Professor CHEKMAN. Thank you for the insightful comments you share with us about publication. As a young scholar myself, I would like to share some of my experience of publishing. When I submitted papers to journals run by professional researchers like PNAS, I find it very difficult to fight with the editor decisions or the reviewer decisions. Even if the reviewer comments came back, obviously, or not correct, I would say so, yeah. But the editor would still be very reluctant to dispute their decisions. Yeah. Then I found that those journals run by professional editors like Nature series, they are more open to such challenges. In fact, 3 of my papers published in those journals initially were not favorably considered. And then after presenting evidences and more arguments, I successfully convinced them that this is an interesting work and you should go forward and send it out to reviewers. Yeah. So maybe from my experience, something I would like to point out is that for young scholars who are not as high profile as those well-established researchers, those journals run by professional editors could still be a great option in some cases. Yeah.
So yeah, well, I'm delighted to hear that you had a favorable experience, but for that one favorable experience, I can give you many others that I've heard from people around the world who have had much less good experiences at these high-profile selective journals run by commercial publishers. And, you know, with any paper, when you send it in to any journal, it's always the luck of the draw, you know, who the editor is, whether it's a professional editor or a scientist. I know when I handle a paper for the PNAS, I ask the reviewers to confer with each other, to share their ideas about the paper and to suggest the key experiments that might need to be done. And very often that gives the author a more informed impression of what needs to be done. and then they come back and it gets done. And so in my case, most often these papers are accepted after revision. So I'm sorry you had a bad experience, but that doesn't, I wouldn't condemn the entire PNAS. It's a very large journal. They publish tens of thousands of papers a year. So you had a bad experience. I'm sorry.
All right, next questions. You already have a question actually about the, the basic research versus the applied research.
Mhm.
Nowadays, the funding agent, even the government, say we need to do research with the impact on the society. So in that case, what would be your advice to our young researcher about, the basic research for publication or the applied research, which may take a long time to realize?
Yeah. Well, of course, this is always a problem. It's true here in the United States as well. And the government officials who are the ones responsible for allocating the funds don't understand that progress depends on basic science. So the case has to be made over and over again. You need to, whoever the government officials who are making these decisions, they need to be educated about the value of basic science. And you can give them the best recent example, which is the origins of the revolution in genome editing with CRISPR-Cas9. The foundation of that breakthrough that has allowed genome editing to be conceived was entirely basic microbiology research that had no obvious practical application whatsoever. There are just a million examples of that. And so the government officials need to hear about this. We need, as scientists, to be organized through our scientific societies to have access to these government officials so that they understand. And I think many of them do. In fact, in the US, there has been a general appreciation by the government of the value of basic science over many years. And it even started with the very first federal funding of research after World War II, where a government advisor to Presidents Eisenhower and Truman wrote a very influential report where he called on the government to recognize the importance of basic investigator-initiated science. And that has generally been embraced. But of course, there are always pressures to do something that seems practical. But in my experience, the best science gets done in the universities at a basic level. And then if you have entrepreneurs in biotechnology and pharmacy, if they have access to this information, they will find a good way to use that. That was true in my work on yeast. I never had any notion of doing anything practical, but what we did eventually led to the use of yeast as a platform to produce clinically important proteins like insulin and hepatitis B vaccine. They're the things that I couldn't have imagined. That's very often the case.
All right, thank you. I think we have the time for one last question. I think the person who raised your hand, please ask the question directly. No? Someone raise your hand in the chat box. If not, then we have one last question from the audience here.
Hello, Professor Randy. Hello, Professor Randy.
Hello.
You mentioned some problems about the paper publication system, like reviewing process or the impact factor. In China, the paper publication is not only about the money reward, it's also about a career like employment or promoting or conferring academic title. It's also close related to paper publication. But it's obviously a burden for those scholars, especially hospital doctors or the college teachers. But would you think it's also a relatively fair evaluation criterion or competition model for them? Because if you cancel these index, would things like unfair competition or casting coach more likely to appear, or like, do you think like is there any better evaluation criterion to replace these?
Yeah, well, you know, people are always trying to think of a simple way of measuring quality. And impact factor was a convenient way to do this. But let me give you an example of why it's a silly number. Here's a tremendously important discovery was made by a fruit fly geneticist many decades ago, a brilliant geneticist by the name of Seymour Benzer at Caltech. He was the first to recognize that it might be possible to understand the circadian rhythm, because even flies have a daily cycle to their life. And so he had a graduate student who isolated a mutation that produces an aperiodic daily cycle for the fruit fly. And they published a paper in the PNAS. That paper eventually led to a revolution in science and led to 3 Nobel Prizes in the last few years, unfortunately, long after Benzer died. But that paper was cited as a reference a total of 10 times over the next 10 years. It was so far ahead of its time that it wasn't recognized for its brilliance. And so if you rely on impact factor to judge the importance, the quality of a paper, you will miss, I think, the most important papers, because the number is calculated on a narrow window. And so these, unfortunately, again, these professional editors, they're employed by these commercial journals to sell magazines. And so they're looking for something that's going to generate interest over a very narrow window of time. So, you know, I'm beating a dead horse here, but there's no substitute in my mind for scholars who understand an area to read and evaluate a paper, or the group of papers where someone is considered for employment in an academic position or for promotion in that position. So instead of just looking at where some scientist publishes his or her work, the group that evaluates the level of scholarship should be charged with reading the papers and judging the work as a scientist, not as a bean counter. It's not easy, but I don't think there's a lazy, easy solution to this. It requires people to actually read the papers and understand the work.
Okay.
So this is a problem with universities, and it's not just in Asia, it's all over the world. Administrators in universities are not well equipped to judge the work of an individual scholar. They have to ask experts, and they have to get honest judgment from experts about the quality of the work, and to base their decisions on those experts and not on journal editors who are selling magazines.
Yeah, thank you. As you mentioned that like it's a problem like about the university. It's also like you mentioned before, like a young scholar should try to publish their paper soon instead of like presuming a higher impact factor or weighting. But as far as I'm concerned, like a young scholar, such like a PhD student, I don't think they get the chance to decide when to publish their paper.
Well, at some level, somebody does, and I'm saying that it's up to the individual, either the mentor or the student, to take a stand at what's important. Is it important to have real peers evaluate your paper, or is it somebody whose business is to sell magazines?
Okay.
All right, it's good. I think we have the PhD student ask the last question. Okay, so it has been really late, I think, in California, right? We have to
Oh, just in time for dinner for
Randy
me.
to go for dinner after the talk, right? So Randy, hopefully we'll see you again in person later this year or next year in Hong Kong.
Yes.
Okay.
I look forward to seeing you, Li Wen.
All right. So please join me to thank Professor Randy SCHEKMAN again. Thank you again, Randy.
Okay. Good night.
Good night. Good night.
Good morning.